This will be our first video of Chapter 4. This unit talks about organic chemistry, so specifically we'll look at Chapters 4 and 5. But we're going to start off with Chapter 4 with carbon, the atom, the element carbon, what that means, and how it lends itself to molecular diversity in all of life. The image in front of you is an adorable, in my opinion, little trilobite beetle. Pretty cool creatures. If you don't know anything about them, go ahead and check them out. Carbon as an overview. So what we're going to see in the next several slides is that carbon is really the backbone for organic life on Earth. Cells have a predominant composition of water. 70 to 95% of them are made up of water, but then the other stuff that makes up that cell has a lot of carbon in it. In fact, it's pretty much based off of all carbon formated or carbon-based compounds. Proteins, carbohydrates, the nucleic acids, both DNA and RNA, a lot of other molecules are all going to have carbon as the composition of their backbones and their base structures. Definition for organic chemistry is going to be the study of those compounds that contain carbon. So what you really want to understand is that when we talk about organics, organic matter, organic chemistry, what we really have to recognize is that there is a predominance of carbon in these compounds. Many of the many of the organic compounds will also contain hydrogen. It's not an end-all be-all requirement the way carbon is, but the vast majority of the ones that we look at in this course, and even the ones that you look at in, Biology 182, 201, 202, they're all going to have a lot of carbon in them. Two ideas from the textbook, vitalism and mechanism. You want to know these definitions. You want to know that vitalism was disproven. You want to know what mechanism means. And really you want to have an idea about the experimental work done by Stanley Miller. Stanley Miller was a scientist in the early 1950s. He did an experiment, and what he wanted to know was his big question. We talk about scientific method and what's your question? What do you want to solve? Well, what Stanley Miller wanted to know was, can organic molecules form under conditions that were believed to be present on early Earth? He wanted to look at that notion that vitalism was wrong, and that he wanted to see if we could actually develop organic molecules without having organisms previously present. This is his experimental design. He started out with this flask over here that had a built-in vapor tube. In this flask, he put water. This flask represented the primeval sea. He heated the primeval sea, or the water, creating a water vapor that then came up here, and that water vapor entered into a second flask. That flask represented, this higher flask up here represented the atmosphere. Also within this flask, he had two electrodes. What he did was he used those electrodes to generate a spark. The spark was designed to represent lightning in the atmosphere. He wanted a synthetic discharge to mimic lightning. And then we had condensation through this condenser here, and what he collected down here was his sample for analysis. Miller's results were interesting, to say the least. What he collected in that sample, when he analyzed it, were a variety of organic molecules. Molecules that are very common in organisms, so things like formaldehyde, hydrogen cyanide, some more complex molecules like amino acids and long chains of hydrocarbons. We'll talk about hydrocarbons in a little bit, but they are chains of carbon and hydrogen. And what Miller concluded was that organic molecules really are the first step of organic life. And based off of his experiment, he believed that they may, he didn't say they were, but he said they may have been synthesized abiotically on early Earth. And that was a big shift from what individuals at the time believed was possible or not possible. Keep in mind that carbon is is easy to enter into so many different bonds and compounds because of its electron configuration. We talked about that in chapter 3 with, the whole shape of molecules are dependent, and all the bonds that we enter into are really truly dependent upon the electrons and the electron configuration. So that really is true. It's going to hold true not just for the inorganics we talked about previously, but here for the organics. Carbon has 4 valence electrons. We call that a tetravalence. Being that it has 4 valence electrons, it also has 4 openings. With those 4 open spaces in the shell, carbon has the ability to enter into 4 significant covalent bonds. Remember, covalent bonds are the strongest of the bond types that we've talked about. So carbon, its most frequent partners are going to be those other 3, the big 4, the other 3 molecules that we talked about in living organisms: hydrogen, oxygen, and nitrogen. So carbon, hydrogen, oxygen, nitrogen. And those guys are going to be the building code that governs how molecules get put together and what they look like. 2 things. If you're in lecture, you know what we do with our whiteboards. If you're in my online course, I'll give you a real quick overview, and then we'll end this video. But the whiteboards are used for students to just do a quick 30, 45, 60-second data dump of things that we've covered. Some really big or key points, maybe a definition here or something like that there. So that's what the whiteboards are used for. If you're studying at home, go ahead and use them. Think about what your answer would be. Here's another whiteboard question, or actually a couple. What is a valence electron, and then what are the valence of the following elements? So I'll just give you these guys. We have 1 opening in this shell, so the valence of hydrogen is 1. In this outer shell on oxygen, there are 2 openings to complete this shell, so oxygen has a valence of 2. Nitrogen has a valence of 3. I have 3 openings here, and carbon with 4 electrons in its valence shell has 4 openings, therefore it has a valence of 4. Those are important to remember as you move through. This looks like probably a good place. Yeah, let's do one more. So remember that carbon atoms can partner with atoms other than hydrogen. So the top example is carbon dioxide, where carbon will double bond with 2 oxygen molecules. Down below we have urea. Let me see if my pen tablet is working. If it's on and working, we can give this a go. So what we'll see here is I'm going to draw over here because I always draw bigger than I intend to. We've got a carbon. Let's double bond it. Remember, I've got 4 bonds that carbon needs to, so we're going to take up 2 there. We're going to take up another one here coming off to this nitrogen. We're going to have another one over here to a nitrogen. Remember that nitrogen has a valence of 3. So right now, nitrogen has 1 bond to the carbon. We're going to give it its 2 little hydrogens. Nobody laugh. It's actually harder to do with your mouse than you might think. Or maybe you all know that. Okay, there we go. So that's going to be urea, and that looks like a good place to stop this video. We'll pick up on our next slide, which is actually going to talk about carbon and the structure of the carbon backbone.